All right. So let's talk about the myofibrils that make up our, or excuse me, the proteins that make up our myofibrils. Three types of proteins or three kinds of proteins are found and make up our myofibrils: contractile, regulatory, and structural. What we talked about for the most part on the last page were contractile proteins. Myosin and actin, which are the major components of the thick and thin filaments. I didn't mention them, but if you notice in parentheses, behind thin filament, it says actin. Behind thick filament, it says myosin. Let's talk myosin first. Myosin functions as a motor protein in all three types of muscles, so it's not just found in skeletal muscle, it's also found in smooth and cardiac. Okay. It is a protein that will push or pull various cellular structures to achieve movement by converting the chemical energy in ATP to mechanical energy of motion. Okay. About 300 myosin molecules form the thick filament. Okay. What that means is if you have 300 of these guys, you're going to form a thick filament. Only myosin is found in the thick filament. I'm going to state that again because it's important. Thick filament is only made up of one protein, and that's myosin, about 300 of them. Each myosin molecule, which is shown right here, is like two golf clubs. Everybody hopefully knows what a golf club looks like. Twisted around each other, where the shafts are twisted, like the the long metal part. All golf club parts are metal now, but the shaft of the golf clubs are twisted around each other. And then the part of a golf club that you hit the ball with are like what these heads are. Okay. Now, within the myosin heads are two major important binding sites. Okay. Actin binding site, guess what it binds with? Actin. Okay. The ATP binding site is where ATP binds and energizes the heads. Okay. What's really cool about the heads is they have a hinge which is different than what we see in a golf club, which is nice and rigid, and you don't want a golf club to swivel like these myosin heads can do. Okay. This hinge is like the hinge of a door, can allow these, and you can kind of see that they're in different positions, to extend themselves like this is doing here, and then flexing themselves, okay, like this one's doing right here. It all relates to energy from the ATP. But what happens is the myosin head will bind to actin here, and then it flexes, it pulls the actin going from this position to this position. Okay? Think of it like taking your wrist and going from an extended straight wrist to flexing the wrist. Okay, so it grabs and pulls the actin, which is what causes this zone of overlap to grow as the sarcomere shortens the z-disc and I'm getting pulled together. Okay, each of these myosin heads can grab and pull the thin filaments. Ultimately, think of it like pulling a rope. If you're pulling something with the rope, you're pulling that rope towards the umbilical. Okay. What's cool about the thick filament is, like I said, there are about 300 molecules of myosin that make it up. Okay, this would be the M line that we showed here on the previous page, or over here on this side of the page. Okay, we have some of those myosin molecules where their heads are on one side and some on the other side, which looks just like what we saw here. Okay, because there are thin filaments on this side of the sarcomere and there's thin filaments on this side. What I really like about this illustration, specifically right here where I'm pointing, is it shows, you kind of have to look hard. See the big red dots and the little yellow dots? Myofibrils are three-dimensional structures. Okay, this is a two-dimensional picture, but there are three-dimensional parts to a myofibril, meaning that there are not just one layer of thick filaments, thin filaments, but there's depth to it. Okay. In fact, one singular thick filament is surrounded by 6 thin filaments, okay, which is kind of hard to show, but I like that it kind of makes an attempt at doing that. Okay. Now, your thin filament is predominantly made, and that's why it's put in parentheses on the previous page, by actin, the main component, as it states right here, of the thin filament. But I'll state this, it's not the only component of the thin filament. Okay. Actin is actually found in 2 forms, G and F. Okay. Think of them like beads on a necklace. Okay. Each bead is a G-actin. And each of those, as you can see here, has a little black dot representing its myosin binding site, which is where those myosin heads attach. Okay. You take those beads and you put them on a strand, and you actually take 2 strands, so like 2 necklaces, and coil them around each other, and you've got an F strand, or an F-actin strand. Okay. And that's the type of, and you can see it fairly well on our picture over here on the right side of the page. That's what we're seeing when we have our completed sarcomere, the F-actin form. Okay. Now, picture below that are 2 other proteins that come together with the actin and make a thin filament. Troponin and tropomyosin, which, as it states, are regulatory proteins because they switch the contraction process off and on. Okay. This set of proteins either will cover, like this, what we see, the myosin binding sites, which inhibits muscle contraction, because myosin has to bind to actin to cause the pulling action to shorten the sarcomeres. But right now the muscle's relaxed, and that's because the tropomyosin is covering those binding sites. But a couple slides ago we said that calcium is released from the sarcoplasmic reticulum when we have an action potential spreading through the muscle fiber. Calcium binds to the troponin molecules. And when it does, the tropomyosin, that looks kind of like a rope, slides out of the way, thus freeing up those binding sites where the myosin can then bind to the actin. Super complicated, a lot more sophisticated than what we really kind of get into in this class. Talk a lot about this in physiology, okay, but not so much in anatomy. I wanted to kind of introduce it to you, but that's about it. Okay. There's a third type of protein in our myofibrils. We call these structural proteins, and they keep the thick and thin filaments in the proper alignment, give the myofibril its elasticity and extensibility, and link the myofibrils to the sarcolemma and extracellular matrix. Of those, the most prominent, and it's the only other protein that was listed on that previous page, is titin. Titin, which gets its name for its large size, is our third most plentiful protein in skeletal muscle, coming in after actin-myosin. And its job, and this is what you'll want to take away from it, it stabilizes the position of myosin by spanning from a Z-disc to an M line. And something very important, and I would highlight this is where that elasticity and extensibility to muscle fibers comes from. Okay. Remember, extensibility, think extend, it allows for stretch. Elasticity allows for recoil. Okay. So it helps with, 2 of those very important properties that we talked about on Tuesday. Dystrophin is a protein that links filaments of the sarcomere to integral membrane proteins in the sarcolemma. Dystrophin sounds a lot like dystrophy. Anybody ever heard dystrophy before? I imagine a lot of you have heard of muscular dystrophy before. Muscular dystrophy, unfortunately, is a condition. Most prominent or most common type of it is, I think it's called Duchenne's, results because of an improper form of dystrophin. And unfortunately, there's really no good way to treat or prevent it. Nebulin wraps around the entire length of each thin filament to anchor the thin filaments to Z discs. Okay. I am not really worried about you knowing any of the illustrations on this page, although I did include this illustration showing you two sarcomeres to illustrate what takes place when you go from a relaxed to partially contracted to maximally contracted, showing you how those thick and thin filaments slide over one another. They've kind of color-coded and boxed off the H zone, I band, A band to illustrate this. Notice the H zone where there's only thick filament starts to get smaller and eventually disappears because the thin filaments are getting pulled closer to the center of the sarcomere. The I band is also starting to get smaller and eventually disappears because the thick filaments are getting closer to the Z discs. What doesn't change in size is the A band. Remember, the A band goes from one side of the thick filament to the other. And that demonstrates that the filaments don't get shorter like my arms when I slid them across one another. What changes is the amount of overlap, which is pretty cool. Okay, like I said, don't worry about the pictures on this page, just the information. Now, table 10.3 is a table that I snagged from the textbook listing off the various levels of organization, which is essentially the exact same thing that I did here. I just kind of did it as we went to kind of keep our head straight. This is a little bit better because it gives you the name, the illustration of it, and the description of it. The only difference is they don't have a segment in here between myofibril and filaments for sarcomeres. They show the sarcomere, but they don't list it as its own. And that's because it's really not a structure. It's just a segment of a structure. But still, you'll want to make sure you can identify, you know, which one's bigger, which one's smaller when it comes to the exam next week. All right. Really quickly, one last slide here to talk about cardiac and smooth muscle before we move on to our next lecture. All right, let's talk cardiac muscle. Cardiac muscle fibers have the exact same arrangement of actin and myosin. So we have the same bands and zones, thick, thin filaments, A band, I band, H zone, all that wonderful stuff that we just talked about that we saw in skeletal muscle fibers. Something unique to cardiac muscle is the presence of intercalated discs, which are irregular transverse thickenings of the sarcolemma connecting the ends of muscle fibers with one another. And these are what the kind of little black zigzags are showing you here in our illustration at the bottom. Within them, we'll talk about these a little bit later on the semester when we talk about the heart, are kind of open doors that allow action potentials to spread from one cell directly into another, which causes those cells to become excited and then contract in what we call unison. We call these open doorways gap junctions. And they're unique. We don't see a lot of those many places in your heart muscle. Cardiac muscle tissue has an endomysium, perimysium, but it lacks an epi. When stimulated by action potentials, cardiac muscle tissue remains contracted 10 to 15 times longer than skeletal muscle because of prolonged delivery of calcium in the sarcoplasm. Mitochondria and cardiac muscle fibers are larger, more numerous, suggesting cardiac muscle fibers prefer to make their ATP aerobically. And like skeletal muscle fibers, cardiac muscle fibers can undergo hypertrophy in response to increased workloads, which explains why a lot of endurance athletes, especially runners and bicyclists, that's terrible, bicyclists have an enlarged heart. That's just a lot of times just called an athlete's heart. Smooth muscle fibers come in two forms, visceral or single unit and multi-unit. We show here visceral and multi-unit. The difference relates to the innervation that exists. With visceral, which is more common, it's found in our skin and tubular arrangements forming parts of the walls of arteries and veins and hollow organs like our stomach, intestines, uterus, and urinary bladder. They are not always directly, each cell that is innervated by a neuron. Okay. They operate a lot like cardiac muscle fibers in that, this cell that's innervated, this one that's innervated. The action potential will stimulate that cell, and then the action potential can spread from cell to cell through those gap junctions. Whereas over here with multi-unit, you notice every single cell has its own connection to a neuron. Okay. That's the big difference. We see a lot less or very few gap junctions between the neighboring cells. Multi-unit is found in the walls of larger arteries, airways to the lungs, erector pili muscles. Remember, those are the muscles in our skin that erect our hair when we get cold or when we get frightened. In the iris, which is a structure that adjusts pupil diameter, and in the ciliary body, adjusting focus of the lens in our eye. Structurally, a single relaxed smooth muscle fiber is 30 to 200 microns in length. Sarcoplasm does contain both thick and thin filaments, but they're not arranged in orderly arranged sarcomeres like we see in both skeletal and cardiac. So we don't see the thick and thin filaments arranged with the zones and bands and all that wonderful stuff for smooth muscle. We also find another filament that we don't find in skeletal and cardiac. It's called intermediate, which is what's shown in our illustration. Kind of look hard enough, you can see it. I'm not really going to zoom in because the picture's not important. I just included them because they were in your textbook. Okay, any questions on this lecture?